that we would start this first session with a few talks that give us sort of a global view of some of the emerging complexities that we've learned over the past few years and their implications for developing therapies for neurodegenerative diseases. So Eliezer was supposed to give 2 talks, one today, one tomorrow, but he's being called to Congress tomorrow, so he has kindly, nicely agreed to merge these talks in terms of his therapeutic talk, which was scheduled for tomorrow, and provide an overview of the current therapies and the implications of copathologies and disease heterogeneity for developing treatments for neurodegenerative diseases. Thanks again for being with us.
Thank you very much. Can you hear me okay? Yes. All right, great. Well, thank you so much to Hilal and the organizers and to you for being here and inviting me to this great meeting. It's one of the first meetings I'm doing in person, so I'm still trying to adapt to the new environment. So it's really great. So as Hilal said, I'm going to give you a little bit more a bird's-eye view of the field, both in terms of copathologies and heterogeneity, and how that is relevant to how we're making decisions in terms of supporting different programs for therapeutics for neurodegenerative disorders. So the framework or the blueprint for everything we're doing at NIH right now is the National Plan to Address Alzheimer's Disease that was signed into law in 2011 and has as its primary goal to develop therapeutics for Alzheimer and related dementias. And related dementias, we include Lewy body dementia, frontotemporal, vascular dementia, and mixed dementias. And the idea is to get to that goal by the year 2025. So we've been very lucky to get quite a bit of support from Congress and increase in funding that have benefited many of you. Many of you are grantees of NIA and NINDS. And we have gone from about $500 million in 2015 to now $3.4 billion a year in funding for these neurodegenerative disorders. Now, during these 5-6 years of increased funding for AD and ADRD, there has been a tremendous progress in the field, and a lot of really very exciting things have happened. I think one of the big things that we've been able to do is to bring together multidisciplinary programs for translation and increasing the pipeline from basic to clinic. We also have been able to bolster all the work that we're doing in genetics and bring that genetics to translation, to functional genomics and translation. Also, we have developed this platform that I'll talk a little bit more later, a multi-omic platform that is publicly available to everybody, AMP-AD, that now has 500 new potential targets through the Agora platform. And we also have developed a number of programs on discovery of novel biomarkers, because if we're going to develop all these new therapeutics, we need obviously new biomarkers. We also need to make these data publicly available, and we're currently increasing the number of shots on goal in terms of clinical trials and targets. And as I will describe, we have now over 300 clinical trials that we're funding. Importantly, many of those trials in care and caregiving, and also importantly, we're also using a lot of the funds to diversify the populations that are being studied, because that was a big problem in the past. Now, one of the things that we have learned during these 5 years, work from a number of different groups, but this particular one is from David Bennett and Julie Schneider, is that Alzheimer's disease, it's quite a heterogeneous disorder, and also that these pathologies could be mixed in nature. So, for example, they have shown that in the brains of cognitively unimpaired individuals, there could be not only Alzheimer's pathology, but also Lewy body pathology, vascular pathology, and combinations of these pathologies. Likewise, in mild cognitive impairment, there is a fraction of those individuals, about 25%, that do not show any pathology, and the other 75% show combinations of these pathologies, being Alzheimer pathology the more common. But in individuals that have actually the diagnosis clinically of probable AD based on dementia, the number of potential combinations of these copathologies it's quite striking. Plus, we have learned in the last few years, and again, this is work by many of you and Brad Hyman, that is, and he's going to talk more about these later. We have come up with new neurodegenerative disorders that actually mimic Alzheimer's disease in the later stages that could present on their own or as copathologies, including part or primary age-related tauopathy, ARTAC or aging-related tau astrogliopathy. This one actually could be shown in combination, as well as late or the limbic predominant age-related TDP-43 encephalopathy. And Virginia here and John, our beloved John, have done a lot of these work in the last several years. And again, these disorders could be presented on their own or in combination with Lewy body, with Alzheimer's pathology, etc. In fact, this is work that has been done with the autopsy material that has been collected for many years from the Alzheimer's centers. And what we see is that in the earlier stages or in the individuals that are looked at the brains in early stages, in their 60s, 70s, as you can see, most of the pathologies, either pure Lewy body pathology or pure Alzheimer pathology, but as individuals age, the mixed pathologies become more common, and actually the combinations of those mixed pathologies becomes more common. So in individuals that are over 80 years old, you can find all kinds of combinations. So this is really important because, of course, comorbidities might be very important in terms of designs of clinical trials, because if you have individuals where you're trying to target just amyloid or A-beta, and they have synuclein, TDP-43, tau, etc., well, you have a problem there. So this also talks to us about the urgency of developing biomarkers to diagnose these comorbidities. Now, what is driving this heterogeneity, or what is driving all these comorbidities? And I think in the field, in general terms, we have thought that probably heterogeneity could be thought in two terms: temporal and phenotypic heterogeneity of the disease, and that probably determinants of these include the aging process. And I think we're going to hear a lot about aging mechanisms in the next few talks. Proteostasis, mitochondrial dysfunction, DNA damage, cell senescence, immune surveillance, and I'll talk a little bit more about that. Such that these might be relevant to the balance between what is resilience versus susceptibility in these neurodegenerative disorders. Of course, also the genetic background is a very important determinant of this heterogeneity. Probably could be 70% of the variant. Understanding this heterogeneity in the context of diverse populations is very important. We know, for example, that African Americans have more combinations that involve vascular dementia, while others have more either AD pathology, Lewy body pathology, TDP-43, etc. And finally, environment, and we're going to hear about viruses and other environmental factors that might also be determinants of this heterogeneity. We have been funding for the last few years a very large Alzheimer's sequencing program. All the data is made publicly available through our NIGAT's database sharing site. And over 50 different loci have been identified, and lipid metabolism, neuroinflammatory, immunity, neuronal synaptic signaling, endocytosis, and mitochondrial function. About 20 of these genes have already been confirmed, and we have now a functional genomics consortia to study these identified genes, how they interact, how they are involved, and how they determine some of these variation, including diverse populations. And we hope that by the year 2025, we will have 100,000 whole genomes available, again, from diverse populations. So now we have all these data on genetics, on aging, an environment that is in the exposome. So how do we disentangle all these data? How do we understand this heterogeneity, both temporal and phenotypic, at all the levels of organization? Because, of course, this is a multi-scale type of problem, and I think we're going to hear probably tomorrow more about the approaches for these multi-scale understanding and modeling of these diseases. And there are a couple of studies that we have funded. One, this sustained approach for temporal heterogeneity using MRI, longitudinal MRI, and trying to understand how in different individuals the disease progresses in different ways. And also this other approach, which is a multi-omic approach that goes from very deep phenotyping at the level of protein, lipids, metabolism, etc., to computational methods to describe different subtypes of the disease. And one of our major efforts in that regard for phenotypic modeling of heterogeneity is the Accelerating Medicines Partnership, or AMP-AD. And AMP-AD has more than 3,000 cases, both control and AD. There is, in this new iteration, a huge effort to have diverse populations, including African American, Latino, having brain, CSF, blood, and doing, as I said, multi-omic approaches, also having a generation of longitudinal immunologic profiling. And all of the data is made publicly available through the AMP-AD knowledge portal. Important to say that this is a very large consortia, over 100 investigators, both public and private partnership. And it has generated very rich multi-omic data from humans. There is a very large number of investigators using it now. And all the novel candidates that have been identified through this platform are available in Agora. And these are just some examples of those targets, like BGF, clustering, and other molecules involved in innate immunity, neuronal polarity, etc. And the important thing about these targets that have been identified by this multi-omic approach is that they serve as nodes that control multiple other functions. So you could actually use interventions at a very high scale molecularly. Now, the investigators of AMP-AD have also used these multi-omic data to try to understand better the subtyping of Alzheimer's disease. And to make a very long story short, in a very complicated paper published in Science Advances in 2021, they discover 3 subtypes, the A, B, and C, the B1, B2, and the C1, C2, depending on which genes and which proteins and metabolic pathways are dysregulated, either in the synaptic pathway, in the immune pathway, or in the protein metabolism pathway, etc. And using this type of modeling and molecular subtyping, they have identified, for example, very important regulatory genes, including, for example, LRP10, myosin. I know we're going to hear more about lipids and membrane proteins, PLP1, etc. So all of these could be used either as targets or biomarkers, trying to develop a better understanding of the phenotypic difference of Alzheimer's disease. But this is, as I said, basically based on expression and phenotypic changes. But we also have to understand the temporal changes of the disease, and not all the forms of Alzheimer's disease temporally are the same. And ADNI, which is our Alzheimer's disease neuroimaging program, have done a fantastic job trying to develop biomarkers and understanding longitudinally the development of the disease. And in collaboration with the Alzheimer Association, NIH, and others have developed this research framework that actually includes biomarkers for amyloid, tau, neurodegeneration, and now novel biomarkers for other structures have been developed, including synapses, vascular, trying to develop for synuclein, TDP-43, inflammation, etc. And essentially classify the individuals by a combination of what is their longitudinal biomarkers changes, their longitudinal imaging changes, their genetic and their clinical data. And one of the studies that have been published is utilizing this technique that I mentioned to you, the sustained approach. And they have identified at least 3 variants of Alzheimer's disease according to the temporal evolution of the changes throughout the nervous system, including the typical, which include the frontotemporal and parietal degeneration, versus the cortical, that is more just like neocortical structures and less involving limbic structures or subcortical structures. And then there is a third type that is the subcortical type, and have calculated the probability according to those subtypes, and there is a pretty good correlation. Now, interestingly, when you combine both the temporal and phenotypic markers from the ADNI data and the AMPD data, what you actually come up with is four subtypes of Alzheimer's disease. The typical one that I just mentioned, but then you have one that is called hippocampal sparing, where there is less temporal cortex limbic system degeneration. These are younger individuals, usually males, ApoE4 non-carriers that have a higher cognitive reserve, lower levels of A beta, but Lewy body pathology with higher levels of phospho-tau and CSF. While, for example, you have the limbic predominant subtype, where it's the opposite. These are older individuals that are ApoE4 positive, have lower CSF tau, but tend to have hippocampal sclerosis with TDP-43 or late that I described before. And the fourth type is this so-called minimal atrophy type. And this correlates also really well with the PET imaging, with longitudinal PET imaging, and the 4 types, again, from a lot of the ADNI data that have been described, that include the so-called posterior limbic, temporal, and temporal sparing. So there is a pretty good correlation there. But I think that a lot more work needs to be done to integrate at the multi-scale all these molecular data with the synaptic, with the networks data, with the imaging data, and with the clinical data. Of course, we have huge voids in the knowledge as to how the multi-scale data translate one thing to another. I mean, I think that we tend to believe that there is a good correlation between the molecular data with the behavioral data, but the truth is that there are a lot of voids or a lot of black boxes. We don't even understand exactly how all these data is encoded synaptically, and how synaptic damage that occurs in Alzheimer's and other neurodegenerative disorders might translate to behavioral deficits and so on. So I think there are a lot of very interesting questions. But the other important thing about this is that I think it helps us developing a trajectory or a blueprint toward developing a precision medicine approach. And I think we're going to hear more about this from Brad Hyman. But I think one thing we should take into consideration is this, and is that I think most of the therapeutics, and now I'm switching to the therapeutics aspect of the talk, have been focused at targeting the protein aggregation problem. And as we all know, and this is work of many of you here, Patrick, Virginia, Jeff, and many of in the Zoom, is a result, this protein aggregation and propagation of proteins is a result of an increased synthesis, increased aggregation, and decreased clearance of these proteins. So the idea has been that, well, maybe either decreasing the synthesis of these proteins or decreasing the aggregation, as we saw from David, or increasing the clearance of these proteins, as we'll hear from some of you, might be the way to go. And I think we have, in general, two approaches when it comes down to targeting protein in these neurodegenerative disorders, which includes targeting intracellular protein aggregates either by reducing expression genetically or aggregation inhibitors or chaperones or increasing clearance, for example, with antibodies, with proteases, autophagy, or targeting the propagating proteins, extracellular proteins, where not only you could actually reduce expression, but you could also, for example, target the receptors to which those protein aggregates that are transmitting are binding to, like mGluR5, TLRs, ephrin, etc. Or you can also interfere with the release of the protein or increase the uptake of the protein with antibodies or other approaches. But of course, that is the proteinopathy component of the disease. Most of these neurodegenerative disorders are aging-related disorders. So there gotta be a relationship between what is happening in terms of the proteinopathy and what is happening in terms of aging mechanisms that might be leading to synaptic damage and eventually to neurodegeneration. So I think as important as it is to understand protein aggregation and dissemination and targeting those protein processes, we also need to target the aging process. And the idea is that if we target either processes like DNA damage, transposable elements, cell senescence, and inflammation in combination with some of the protein aggregation processes, maybe we might develop better therapeutics. There are some really interesting studies that have been done both with some drugs, anti-senescence drug targeting cell senescence. In the top is an APP transgenic model showing that deleting with a combination of dasatinib and quercetin, which are anti-senolytic type of drugs, we can diminish a lot of the behavioral pathology and pathological changes in a model of AD, and likewise targeting genetically senescent cells in a tau transgenic model. It has been shown that a lot of the pathology and behavioral deficits can be reversed. So we have now some clinical trials in the field involving cell senescence. There are other aging pathways that could be targeted. For example, the AMP kinase signaling pathway with metformin, and we also are supporting clinical trials in that regard. The other one is the DNA damage, and there is interest on some investigators, especially at Scripps and other places, of targeting DNA damage with anti-retroviral drugs, and also the autophagy pathway with the mTOR inhibitors, and we're also funding some clinical trials in that regard. But in general, we're really funding the whole spectrum of the drug development pathway, including the so-called Valley of Death, going all the way from target validation to assay development, proof of concept, etc., and enabling to phase 1, phase 2, phase 3. And we just issue a couple of new programs, especially for these so-called drug discovery of novel AD targets that are not amyloid, but are some of these novel targets. And I think it's something very exciting. And this shows you our funding throughout the years for different targets in preclinical and clinical studies. And in the light blue is A-beta as a target. And as you can see, over the years, there is less and less research we're funding just on A-beta as a target, and there is more on other areas. Tau is the next one, but a lot also new work going on in inflammation, synaptic plasticity, vasculature, neurogenesis, ApoE, neurotransmitter receptors, cell death, etc. So it's a very diversified portfolio that we have. And I mean, this portfolio includes, again, I mean, we still have a lot of targets that involve A-beta therapeutics, but also including synaptic plasticity, and as I mentioned, tau, neuroinflammation, cell death, ApoE, and other targets. In fact, this is a classical paper that Jeff Cummings published every year about all the clinical trials that are in phase 1, phase 2, phase 3 for AD, ADRD, and all the different targets. And the ones that you see in circle in red, those are NIH-funded clinical trials, so quite a few of them are funded by us. As I mentioned before, we have over 300 clinical trials that we're funding. Of them, 72 are pharmacological interventions. Amyloid still the most common, but also followed by inflammation, synapses, vasculature, circadian rhythm, and some of the other targets that I mentioned. But we also have a lot of non-pharmacological trials, 120, that includes exercise, cognitive training, neurostimulation, or combinations. And the most abundant category is for dementia care and caregiving, 155, where, for example, what we're trying to do is improve caregiving health or intervention in the form of care setting or interventions in the home and caregivers, et cetera. So a lot of different trials and interventions. Just some examples, for example, there is this drug that was developed by Frank Longo, LM11A31, that just finished the phase 2 clinical trial, which is a drug, a small molecule that targets the P75 receptor, a neurotrophin receptor, in a double placebo trial. It didn't hit the endpoints on the behavior that they were expecting, but there were some really very good results in biomarkers, especially synaptic biomarkers in CSF and some of the PET imaging and so on. Another really interesting drug is this drug, levetiracetam, which is an SV2A agonist. It's an anti-epileptic drug, and Leonard Mookie has shown before in animal models that this drug decreases network dysfunction, synaptic dysfunction, and now we're funding a phase 3 clinical trial from Agent Bio with a low-level, low-dose formulation of levetiracetam in individuals with MCI. And I mean, we're very hopeful about this trial. But I mean, we have a number of other trials we're funding that will be reported at different time points. We just had our Colombia trial reporting. These are individuals with genetic Alzheimer's disease with a presenilin mutation treated with crenezumab, and unfortunately didn't meeting the clinical endpoints, but meeting some of the biomarker endpoints. But as I said, we also have a lot of non-pharmacological trials coming up, and a number of phase 3 trials coming up. This particular trial is one of the few that have actually shown something. This is the SPRINT MIND trial, which is intensive reduction of blood pressure. And this trial actually show a reduced conversion to dementia of individuals with MCI with a very aggressive reduction of hypertension. And again, this is the only trial so far where we have seen activity. As you know, many of the anti-amyloid immunotherapy trials, including those from Eli Lilly with solanezumab, from Roche with crenezumab, and others have failed. And as I just mentioned, we recently had the report of the DIAN-TU trial and the Colombia trial. And what is different about these trials that we were funding is that they were in individuals in preclinical Alzheimer's disease. The individuals in the Colombia trial, actually, they were started to be treated when they were in their 20s and 30s, before they actually had clinical disease. Even some of them didn't even have amyloid deposition in their brains yet. So, I mean, we still have one other trial coming up, which is the A4 trial. This is a Rhesus Sperling trial. It's a secondary prevention trial. So these are individuals that are amyloid positive by PET, but clinically unimpaired, and they were treated with solanezumab at a high dose. Of course, a lot of these trials had the problem that we had to go through COVID, and the trial had to be suspended and then restarted, et cetera, all these crazy things that happened. But this trial, it will be reporting soon, and I mean, there is also great expectation to hear more about what will happen with this trial. So the other important thing about this drug development pipeline that we've been developing, as I mentioned, a lot of the target development based on AMP-AD has also been to support the translation of a lot of these new unconventional targets into the clinic. So we've been developing a lot of open science infrastructure that enables a lot of these drug development, including the Treat AD centers that were just recently funded and the Model AD centers that have been funded for quite a while. The Treat AD centers are also these partnerships where all the targets that have been developed by others or by the AMP-AD consortia could then be further developed with in vitro testing and all kinds of other assays. And the Model AD uses all this information to develop new knock-in models of late-onset Alzheimer's disease, utilizing all the data from the ADSP that I mentioned to you. They have developed knock-ins with TREM2, ApoE, BIN1, Tau, APP, and many other genes. There are over 50 of them. All of this is available to everybody. And it's really a consortia that involves also consortia for vascular dementia, for MUB2, for resilience, neuropsychiatric disorders, and as I mentioned, animal models and centers for new medicine. So just to conclude, and this is my last slide, I think we definitely need to increase the sharing of all this data, the valuable data that you guys are developing, and I think that's going to bolster our development of new drugs, new targets, and new biomarkers. I mean, we still believe that A-beta and Tau remain important targets for intervention. I mean, we'll see what other primary prevention and secondary trials will show, but I think still targets for prevention, I think they are still valuable. And of course, the heterogeneity of the disease makes us think that probably we need to hit different targets and also develop combinatorial therapies. We're not only targeting protein, but also aging and non-pharmacological. And a lot of the new drug development, I think, will be coming from these multi-omic approaches, and I'm sure many of you will hear a lot more. And I think that emphasizes the importance of having a diversified portfolio and moving forward toward precision medicine and toward combinatory therapeutics. And thank you again. And this is the places where you can contact us if you have any questions or need any help. Thank you.
Thank you. We have time for questions. I just wanted to set the stage for questions. You know, really the main purpose of this meeting is to have discussions to raise difficult questions, have discussions on uncomfortable topic, challenge hypotheses, and hold assumptions. So please feel free. I think everybody understands that's the goal here. Feel free to raise and question things. So I'll start.
Thanks. This was very nice talk. But I have a question. So in Werner syndrome, which is a premature aging syndrome, which has a lot of the phenotypes of aging, there doesn't seem to be any connection with Alzheimer's disease. And so I'm just asking a kind of challenging question. Is it possible that aging just takes time, and so does Alzheimer's disease, they're just co-progressing rather than being connected?
Yeah, no, I think it's a great question. And I think it depends on the pathway through which you enter aging, if it's going to crisscross or not with the pathogenesis of Alzheimer's disease. I think, I mean, what we have seen is that I think P21, P16 it's really very important for the type of cell senescence that crosstalk with what we see in terms of protein aggregation and dissemination in AD-ADRD. I can't remember right now what is the Werner syndrome target, if it's P53 or ATM.
It's a helicase. The Werner helicase.
Oh. So, I mean, probably it is not in the same pathway. Now, I mean, there are other, like progeria and other aging mimic disorders where, I mean, definitely there is degenerative pathology in the nervous system, not necessarily with protein aggregation or not, but I think that might also depend on the time and other genetic variants, resilience factors versus non-resilience factors. But at least for AD, we need to go on that P16, P21 signaling pathway.
Can you hear me okay? Okay. Hi, this is Ignacio from CHDI. I work in the Huntington's field, so maybe some of these questions are less than perfect in terms of education. But one of the problems that we face is with the preclinical evaluation of drugs is the fact that none of the rodent models of HD have progressive neurodegeneration. I don't know how it is in other indications, but does the NIH plan to finance primate models that can advance the evaluation of therapeutics? That's question one. Question two is, it also strikes me that people go a lot in Alzheimer's and Parkinson's from CSF quantification or imaging of amyloid to a clinical scale impact without understanding circuitry changes that may be speaking that the drug is having the intended effect pharmacologically, but it's not changing the progression of the disease. Is there a link between the neuroimaging initiative and trying to test this clinically in the context of existing therapeutics? Thanks.
Sure. Yeah, no, thank you. If I understand well your first question, we do fund a lot of private industry, actually. I mean, some of these, was that question private, you meant private industry? Or?
Private model.
Primate.
Oh, primates, yes, yes, yes.
Genetically engineered primates.
Yes. In fact, we have a funding opportunity out on non-human primates modeling, and also on not only non-human primates, but also other models that are developed spontaneously, AD-like pathology. Definitely, there is a lot of interest on that, and definitely there is now funding opportunities available. But even without the funding opportunities, we're very interested. And by the way, also Austin Yang is here today, also from NIH, and could help. And the second question about it's really it's FDA. I mean, the endpoints that goes into a lot of those clinical trials depends on basically what FDA accept on the initial stages of the negotiation. And I mean, they always request these clinical endpoints as the primary outcome. But now they have started to become interested on accepting some of the biomarker endpoints, particularly the PET imaging. We're hoping that now that we have all these new plasma biomarkers, those will also be part of it. But you're absolutely right. I think we need to have also synaptic network level type of biomarkers. And that trial that I mentioned with Agent Bio, with levetiracetam, they are using a functional MRI and EEG as an outcome measure. But I think it needs to be worked out with FDA. Yeah.
Eliezer, I'm curious from your bird's eye view of things, what you think about the role of cerebral amyloid angiopathy and dementia in general. I mean, 5 years ago, I think there was a very small group of people working on that. Now it seems to be more mainstream. And so what's the view?
No, I think you're right. There has been a dramatic shift, and also a dramatic shift in the interest on vascular pathology in general, just as vascular amyloid triggers damage and bleeding, ischemia, etc., but also as a target, developing animal models, developing therapeutics. Yeah, I think it's extraordinarily important. Also in relationship with diverse populations to whatever, there is evidence that there might be more vascular damage in Hispanic and African Americans that might be linked to A beta 40 rather than 42. So definitely it's something that we need to think about.
Eliezer, so one thing I wasn't certain and I wasn't remembering is that of all of the clinical trials that have been ongoing, particularly those that are using antibodies to reduce beta amyloid or tau, so were there, the data, did they show reduction but no efficacy, or there's no reduction and no efficacy?
No, you're right. Most of them have shown on the biomarkers reduction of amyloid,
Right.
but no
No
clinical
efficacy
changes.
changes.
Well, of course, with the exception of this very controversial Biogen trial with aducanumab that, I mean, they
Biogen trial showed reduction in no efficacy.
Well,
Of amyloid in no efficacy.
that's why I preface
No
with
correlation
controversial.
between reduction of amyloid in that trial and efficacy. None at all.
Were there any trial
That's on
that
page
showed any
68.
correlation whatsoever?
There's no
Okay.
correlation in any trial.
Yeah. Okay.
There was a whisper of efficacy, of course, but it doesn't correlate at all to amyloid.
Okay,
Okay.
we have
So,
two quick.
Eliezer, I'll be over here. So I really enjoyed your presentation. I mean, you make some very, very important points, particularly the fact that there's so much copathology in these patients that doing clinical trials is going to be very challenging when you have multiple pathologies in these individuals. That's hard to sort out. But, you can learn a lot from rare diseases and where the pathology is a little bit more consistent. And I was wondering along those lines what NIH is funding for, let's say, Down syndrome and some of the others. And the second thing is there any emphasis on using gene therapies for targeting known genetic causes of dementia?
Yeah, no, definitely Down syndrome, it's very, very important. We actually have got from Congress additional funds for Down syndrome, and we have a longitudinal study similar to ADNI for Down syndrome called ABCDS, doing all the biomarkers on Down syndrome, and they already have started a vaccine clinical trial targeting A beta, but instead of with infusion of antibodies with an actual vaccine. And there are other ideas about intervention with anti-tau antibodies, but somewhat controversial, but definitely Down syndrome is quite crucial.
One final question for Pete.
Eliezer, I
Peter.
just want to make you squirm, and I hope you forgive me, but I guess you're going to Congress tomorrow. I mean, what do you tell them about the spending in Alzheimer's research, and how are we going to disprove the amyloid hypothesis if we don't accept that reduction, that none of these things work? How, I mean, is there a description? I mean, I was taught that the only way a hypothesis has to be disprovable. So what is the disproof of the amyloid hypothesis if not lowering plaque by imaging, lowering A beta secretion by secretases? All of them fail to produce any clinical benefit in any trial. And so why the continued massive spending?
So how do I squirm? Yeah.
Well, I mean,
first of
if I were involved in that spending of that money, I would really question
But
why,
no, but I
when
think,
do we get to the
I
end?
think I show you very clear. We have a very diversified portfolio. I mean, if anything, A beta in terms of clinical studies is going down. We're just seeing the tail end of A beta, and now we're seeing an explosion on anti-inflammatory, anti-tau, and other things. But I think to your first question, to me, I mean, I think different people might have different answers, but to me, the killer experiment would have to be to either shut down APP production or shut down completely APP synthesis in a primary prevention trial. Because most of these trials that have failed are in individuals with mild to moderate AD. The more recent ones, like the Columbia trial and the Diane, were slightly earlier, but still targeting A beta and all kinds of different things. I think you need to shut down APP production, either gene therapy or other approaches, in the very, very early stages, preclinical, pre-amyloid. And we had a trial like that, and it was going to be done with a BACE inhibitor, because it was thought, well, with the BACE inhibitor, you can really shut down ABP or A-beta production very early on. But then the BACE inhibitor turned out to be super toxic. So that's why it has to be discontinued.
ABP is probably even a worse target. I mean, I think that theoretically, I like that. Because it
But
gets you
just
back to the
theoretically,
original,
right.
And it gets away from this fixation on amyloid. But
Yeah.
to APP, but APP as a target, I think that scares the hell
We
out
can
of me.
All right.
continue at the end. Thank you very much.
Thank you.
For a beautiful talk. Thank you.